So once again, good afternoon, class. We are, I think today's 29th, and I think our next class would be, this is March, a month of 31st. So we'll be meeting Thursday, and then we are going into April vacation. So April would be a busy month. Good, and so in April, before we go for the break, we will have a short quiz on stem cells, which is, I believe, is on 5th April, maybe 6 to 10 questions. And then there will be a midterm cumulative, everything. So my advice generally for midterm prep is, or any exam prep, is to go through your study guides. Study guide that, or assignment that you have done, submitted, that will benefit you a lot. And but in case if you want me to review then something, then do let me know do let me know, day after tomorrow on Thursday, so that I can pick up that topic going into next week. So I mentioned to you that stem cells are undifferentiated cells which have capacity to replicate and to differentiate. But replication is and differentiation for therapeutic purposes, it is based upon the environment we provide. So so if the cells have to be brain cells, then the conditions for growing brain cells from a cell, epithelial cell, the conditions will be very different as compared to skin cells, right? So, and the power of stem cell is that because of its growth capacity and differentiation capacity, it can serve as a therapeutic agent for many diseases. And one of the diseases that I have mentioned that I have uploaded on the Blackboard is that stem cells and cartilage regeneration. So this particular gene, TRPV4 gene, if it is given to a person maybe with chronic arthritis, then this gene can trigger the development of cartilage. And as we know that cartilage is important for the mobility of our knee or ankle, or and that is the major major site of degeneration due to arthritis or any ligament break or something. So so this is one of the latest one of the close by latest publication. It was published in August 24, 2021. So TRP4 gene is essential for development of cartilage or cartilage growth. So another paper that I have put on the Blackboard that I've uploaded on the Blackboard is stem cell, another stem cell publication on embryonic stem cells, a historical overview, so that you can read, as and when you get time, and look at what's going on. It's a general overview, but it's an exhaustive overview. If you're interested, then you can definitely study. The third paper that I have uploaded on the Blackboard on stem cell is stem cell and undergraduate. So stem cell and society, and undergrad course exploring intersection among science, religion, and law. So stem cell is a topic because I mentioned to you in the last class, because to work with human embryonic cells, we need to get experimental stem cells from human embryo, 5th day blastocyst, inner mass cells, for human, to work on embryonic stem cells, human. We discussed this pluripotency, multipotency, oligopotency. These definitions are important because that's how you understand how the potential of a cell could be. So, totipotent, zygote, pluripotent, the three germ layers, and then multipotent, one of the arm of pluripotent mesoderm that develops into bone marrow, and bone marrow can develop into blood cells. But blood cells you can classify into red blood cells, immune fighting cells, and basophil, neutrophils, eosinophils, mast cells, platelet. So you can see how, so just imagine that in our bone marrow, how many different cells are being generated at 24 hours time. And these cells are at the forefront of fighting infection. You can see that there are some that, that suddenly the temperature has, dropped so much. So a lot of people are getting sick, and most of the people are having sore throat. So they are scared that maybe the COVID is coming or something. So these blood cells, lymphocytes, they are very important for combating viral or bacterial infection. So stem cells, replicate and differentiate, but it needs a right climate to develop into brain cells. It needs a different stimulation, different growth factors, different vitamins, different interleukins, so on and so forth. Similarly, the red blood cells, right? So let's say if there is a patient who has sickle cell anemia and is not able to make healthy RBC, then if the bone marrow transplant happens successfully, then the new bone marrow precursor cells can make a healthy RBC. So these are the definitions of stem cells. We covered that blastocyst fifth day, and inner cell mass are the human embryonic stem cells. So I would skip few slides, and I would come to this slide and discuss that inside the bone marrow there can be three different kind of stem cells. How do these differ? These differ on the basis of receptor. What is a receptor? Receptor could be a protein or glycoprotein or lipoprotein molecule on the surface, just like we have cell phones, and cell phones can receive signals, and there are different varieties of cell phone. So let's say in hematopoietic stem cells, the marker is CD50 plus. So now why these markers are, right? So let's say I have a unique social security number or driver license number. So when I show up and on international travel at JFK airport, they will look at those numbers, compare my background check, whatever they want to do. So likewise, these cells also have unique social security number. These are called as CD. CD stands for clustered differentiation. So hematopoietic stem cells, which could give rise to blood cells, they are CD150 positive. Multipotential progenitor cells, they are CD244 positive. So these CD is clustered differentiation representing a type of cells, specific type of cells, and number like CD1, CD2, CD3. So while I was doing my PhD, these things were emerging, the nomenclature, because nomenclature is important to communicate at an international level, at a global level, so that we can speak the same language. So at that point of time, CD1 was developed, CD2 was named, CD3 was T cells, CD4 was CD helper cell, CD8 was cytotoxic cells, CD19 was B cells. So these nomenclatures were coming up to streamline the immunological cells. Then you have the leukemic stem cells, right? Leukemic stem cells, they have two positive. So CD8 48 positive and 244 positive. So if we are doing an experiment with a patient who is leukemic, and you want to put a a gene or edit a gene or to treat those cells with chemotherapy compounds, then of course you don't want to treat these cells. You don't want to treat these cells because these are normal cells. You only want disease-specific cells. So we need to purify these cells from the bone marrow or from the blood cells, and then treat these cells with chemotherapy molecule. And then once either these cells are modified, they stop growing fast, or they are genetically edited because the gene that is contributing to mutation, to leukemic, that can be rectified. So these then, after rectification, these can be put back into the patient. So why to use patient cells for treatment? Because it prevents the chances of rejection, right? So that's how the stem cell um, therapy works. Right? So cells are the diseased cells are withdrawn from the patient, and then they are taken to the lab. And these labs are very sophisticated for maintaining sterility, because let's say if there's a bacterial infection or viral infection, and then sometimes bacteria don't grow fast and they grow slowly. So it is possible that if the patient's sample, which has been treated, and there are slow growing bacteria, so it will grow very fast in human body, because human body has ideal environment, pH 7, lot of nutrients. So then patients will go into septicemia, infection, and then it will be very difficult. So patient cells, treatment, and put back into patient venously, and that's how the treatment. And so this slide, I discussed about adult stem cells. So there, of course, there are points, there are issues in the science which are controversial. This has been historical. But science is like, when once one door becomes controversial, they try to open a new door, but that's again comes with its own challenges. So adult cells are not, so the stem cells, human embryonic stem cells, are undifferentiated naive cells, because they're coming from embryo that has not been exposed to environment. But adult stem cells, they are exposed, they're rigid, very difficult to grow in the lab, very difficult to manipulate. And by the time when a person becomes adult, the hypothesis is that every organ, matured organ, has a stem cells, as compared to in a embryo, where the fifth day embryo has inner cell mass, which is human embryonic cells. So look at these terminologies and difference between adult cells and I mentioned to you that for growing, one of the ways a nutrient can be provided to stem cells is to grow these stem cells in presence of mouse cells. These mouse cells will provide growth factors. If you remember your 202 class, or autocrine, paracrine way of controlling the secretion. So stem cells essentially started with 1975, 1980, and then sheep cloned from Dolly sheep was cloned, and unfortunately that sheep died, and that sheep is in the museum, I believe, in Europe, in Edinburgh. So also the first human embryonic stem cells was harvested, and in 1995, when Bill Clinton was president, and then after Bill Clinton, when George Bush came, then his policies were different as compared to Bill Clinton. Then after George Bush, President Obama came, his policies were different. So you can read this history chart. It is so fascinating the way science grows in front of us, right? So and first clinical trial for spinal cord injury patients, and first production of human stem cells, embryonic stem cells from fetal cells using therapeutic cloning. First clinical trial, so it's going on. So this is a similar slide. And I also mentioned to you about induced. So what have we talked till now? We talked about human embryonic stem cells. We talked about adult stem cells. We talked about induced pluripotent cells where the genes are so 4 genes could be inserted into mouse fibroblast, and these fibroblasts then they can develop into primary induced pluripotent cells. And so these 4 genes are OCT4, SOX2, KLF4, CMYC. Also, human cord blood cells, they also have potential to be used as stem cells, but they still, these are developing areas of biomedical research. So and so different, as I mentioned to you while I was teaching, just mentioned about the article on the blackboard, a gene that is specific for cartilagenesis, development of cartilage. So these genes, KLF4, SOX2, CMYC, NANOG, OCT34, LIN2, in various combination and permutation, they can give rise to either cardiomyocyte, adipocytes, neural cells, hematopoietic progenitor cells, pancreatic beta cells. Now, why do we need pancreatic beta cells, stem cells? Because beta cells are those cells inside pancreas which produces or which secretes insulin. And insulin, as you know, is important for diabetic patients. So sometimes the pancreas are so much atrophied, the tissue is so much damaged or sick or inflamed that it's very hard to control a patient by just, and how much a patient can take insulin, right? Sugar could shoot up, none of the medicines could control. So what remains is stem cells. So patients' cells could be taken out from the same patients, and that stem cells could be treated with one of those genes in various permutation and combination. And then once the cells develop into pancreatic B cells, this can be injected. So the program, the logic makes sense, but there are challenges, and special challenge is to control the cell fate, right? It's very difficult to control the cell fate that, okay, you're treating the patient cells with particular gene and it should develop into pancreatic B cells. It does not develop. It sometimes gives very hard time. And even if it develops, you want to repeat the experiment to make sure that you are doing the right thing. It doesn't work. That's the nature of and the challenge of working with stem cells. So it's a very exciting area and a lot of new work to do in this area. So this is also, I have put very same concept to transform this or transduce these cells with these 4 genes, OCT4, SOX2, CMYC, and KLF4, and develop into iPS cells. These iPS cells could be developed into various tissues or organs, and they can be developed into human disease models. For example, so let's say if you take these cells and you insert these genes, and if these genes also express those 2 CD markers, that is for leukemic stem cells, then these cells will develop into leukemic cells. And these leukemic cells could be used for developing new medicines. So what are we talking here? We are talking about very, very simple things: stem cells, zygote, and these stem cells could be. So knowledge of cellular biology is very important in terms of, because every day you have to see the microscope, you have to look at the behavior of the cells. So so you need fundamental knowledge of cell biology and genetic engineering. So what these things are: OCT3, KLF4, SOX2, CMYC. So they all are transcription factors. What are transcription factors? Transcription factors are those proteins that, of course, by the name it suggests, transcription factors will participate in transcription, right? Will participate. But what happens if it's participating in transcription? Then these proteins, they work with RNA polymerase 2, which is the protein that is involved in transcription. So transcription factors work with RNA polymerase 2 to regulate transcription. So RNA polymerase, what it does, it binds to the promoter, right, start site. And what these do, they also bind in the vicinity of RNA polymerase. Now, these four genes, they might be expressing at different level. OCT4 could be more, this could be less, this could be more. In one situation, this could be more, and they can regulate expression of certain gene. So the nature, the molecular interaction between these four genes will determine the outcome of the transcription and translation and the cell fate, whether it will develop into pancreas or neuronal cells or skin cells or adipocytes. So, stem cells is also very successful in treatment of eye, where there is a macular degeneration. So, patient's cells can be taken. So, stem cells are removed from the patient's good eye and placed on a sheet of amniotic membrane. The cells are immersed in a solution of patient's blood, insulin, and hydrocortisone. So, everything from the patient. So, patient cells are taken, they are treated in the lab for regeneration, and same insulin is here hormone, hydrocortisone steroid. So these cells, they grow here and then they're transplanted back to the defective eye, right? And there's a cornea degeneration. And yeah, so same thing, stem cells are very important for in neurodegeneration and Alzheimer's disease, because these in patients where Alzheimer starts there, they starts it's memory loss and functional loss. So stem cells could be very, very helpful here. Then same stem cells which are important for development of normal things, normal organ can become cancerous, and they can keep dividing, and if the chemotherapy is not working, then it has a fatal effect. So the hypothesis is that cancers or a tumor, right? A tumor is growing inside the patient's body. So a tumor starts as a simple outgrowth of cells, then it grows into a polyp, and then a tumor, then this tumor disseminates into the whole body of the patient, and it depends what stage it is, right? So, and while it is happening, there are mutations. So the hypothesis is that this cancer, given this so much of metastatic potential of a cancer cell or tumor cell, the hypothesis is that there is a cancerous stem cells also. So which has a very fast renewal rate or replication rate. So scientists are trying to work on cancerous stem cells. So what happens? The problem with cancerous stem cells is that they stay dormant, right? So one of the concept that is coming up is dormancy, cellular dormancy. So cells are fine, normal. Cells, what happens when they're not functional? They go and rest, and if they become abnormal, they can cause disease, and they can stay dormant. So breaking cancer dormancy means stay silent and then spring back into action when there is a right condition. So when a person is immunocompromised, these cancerous stem cells can come into action and start growing. So that's also a very important part. So so this was about stem cells. Do you have any questions? So the next quiz, which is, I believe I announced on April 5th, will be only stem cells, and there will be 5 to 6 questions. And then I believe on April 14th, you have your midterm, which will be cumulative, multiple choice questions. And for that, if you're asking for my advice, focus on your study guide and of course, recording and slides. Do you have any questions?
No.
Okay. So, yes.
You took attendance, Professor?
Yeah, we can. Right now, I'm trying to teach. We can take care of attendance later on.
Okay.
Just be patient.
Professor.
Yes.
So did you put up a study guide for the midterm, or is just going through everything that we covered throughout
Everything.
the time?
Study guides are assignments.
Oh, okay, okay. Well, and the stem cell quiz.
Mm-hmm.
Is it going to come out, like questions are going to come out from what we did last week, Tuesday and today on stem cells?
Yes.
Okay, and you said it's 5 questions. Is it 5? Like,
No, I said 5 to 10
questions?
questions. I said 5 to 10 questions.
Okay, all right. Is it multiple choice or written out?
Multiple choice questions.
Okay.
Any other questions? Okay. So I am beginning the new chapter, which is on gene regulation and gene manipulation. So why I am talking about gene regulation is that we just talked about stem cells, right? And we talked about various cluster differentiation, which could be important for targeting particular leukemic cells or cancer stem cells. Now, just I'm trying to open the chapter. Yeah. Now, of course, you cannot isolate this protein from human cells. We have to take help of bacteria, right? And we have talked at length about the role of bacteria in recombinant DNA technology. But this chapter is very, very simple. This chapter, what it introduces or what it teaches is that how come a bacterial cell that is containing recombinant protein or recombinant DNA, how does it scale up the production of protein, right? So let's say flu season, this is spring, flu season might be coming. So these companies will scale up the production of flu vaccine. So in contrast to the COVID-19 vaccine, which is mRNA based, flu vaccine is protein based. So that will be also be made inside the bacterial cell. So how do you scale up, right? The bacterial cell. We cannot produce vaccine inside human body. It will be unethical. So it comes back to bacteria again for scaling up the protein. So let's look at what is the system that is utilized by these companies, right, to scale up the production of protein. So human genes which express protein is a mosaic of introns and exons, and the introns are removed after primary transcript is generated, and the exons they come together, ligate, and then the protein expression occurs in ribosome. Right? But bacteria has a polycistronic RNA. Polycistronic means in contrast to the eukaryotes, which has introns and exons, bacteria bacterial protein is made in one transcript, but this transcript has various cistrons, cistron 1, cistron 2, cistron 3, or n number of cistrons. So these cistrons don't confuse with exons. There is no introns in the bacterial genes which express protein. These cistrons are next to each other, but these cistrons, they code for different proteins, and different proteins do different function upon translation. So human system, so human DNA that codes for protein, I repeat, human DNA that codes for protein is called as DNA coding genes, right? Because not all 100% of human DNA makes protein. Only 2 to 3% of human DNA makes protein, right? So rest does the regulatory part. So human is intron exons, bacteria has cistrons. There are no introns. So this particular thing is being utilized by the companies to scale up protein. So see here, there is one promoter. Promoter is a site where RNA polymerase binds. And RNA polymerase once binds, it goes through 5' to 3', and they make polycistronic RNA, and those RNA can either they are made independently, cistron 1 RNA, cistron 2 RNA, mRNA, cistron N RNA, and they make individual protein. So let's look at the real biological system in bacteria. The real biological example is lactose system. Lactose is called as lac operon. So, if you see here, LAC, this is written in italics. This is written in italics because whenever you write a gene name, you italicized. So let's say you're doing mRNA and you're talking about some gene, then you italicize this in italics. If you don't italicize, it means that you're talking about a protein. So these are standard nomenclature in the field of biology. When you're writing somebody's name, scientific name, like Escherichia coli, E. coli, you write it in italics. So this is lac operon. It is called lac operon because it is a gene which expresses those proteins that are involved in lac metabolism. So lactose, right, is one of the staple bacterial diet when glucose is absent, right? So when glucose is present, then they don't need to work. But sometimes glucose is not available, lactose is there. So presence and absence of lactose in bacterial body, presence and absence of lactose inside the bacterial body can impact an operon. What is an operon? Operon is a gene. It could be a multi-cistronic gene that can code for various proteins. So a lac operon would be made up of promoter or regulator, then promoter where the RNA polymerase bind, and the 3 genes, LAC-Z that codes for beta-galactosidase, LAC-Y that codes for permease, and LAC-A that codes for acetyltransferase or transacetylase. So in the absence of lactose, this doesn't need to operate, right? So what happens? This doesn't operate, right? Because lactose is absent. So who controls this, that gene doesn't function? So there is a protein. So these are the 3 genes. Upstream of 3 gene is lac promoter, where RNA polymerase bind. Upstream is gene regulator. So this gene regulator, regulator gene rather, is called as LAC-I. They make repressor. Repressor negatively influences the transcription. Repressor is put a break, stop sign, like shut down. And what does this repressor does? It goes and binds to the RNA polymerase binding site and blocks it. So if repressor is there, RNA polymerase cannot bind, so no transcription, simple. But in presence of lactose, right, where bacteria has to utilize lactose because there's nothing to eat, they need to generate energy. So lactose comes into picture. Now, what happens? There's an inducer called as allolactose that binds to the repressor, and repressor cannot function. Allolactose serves as a activator, inducer that binds to the repressor, and repressor cannot function. But that gives some chance for the activator to go and bind with RNA polymerase, and these genes are expressed. So how I teach this, that this is brake and this is accelerator. Brake is put in, is exercised by repressor. Accelerator of a car is inducer. And that's because the repressor is inhibited by molecule. Now this activator that activates bacterial transcription has developed into a product. And that product, if it is added to bacterial culture, then bacteria starts making protein a lot. So simple lac operand model serves as a model inside a biotech company to scale up the protein production. Right? And this is a very simplified picture, stop sign, and then activity or activation. Now this is a lactose, right? This is a lactose, right? And so lactose is not that biologically stable. So scientists, they found this molecule called as isopropyl thiocharacteriside, IPTG. They are the inducer, and they express protein, like it just bump up 100 times protein production. I have worked with this compound in my postdoc, and if you want to scale up a protein production, you add into the bacterial culture and you will see that a big band of protein is made. So for example, GFP can be induced in a bacterial in a system. So just a basic difference between eukaryotes and prokaryotes. Primary transcript made up of exons and introns, and then introns are removed by splicing, then processing. Before that, I was talking about mRNA. Then mRNA is modified at cap and tail, and then cytosolic, on the ribosome, and this protein is expressed. So in eukaryotes, gene is regulated, right? More complicated way as compared to bacteria, because human body is complex. Right? It takes 9 months to develop, and the growing embryo from day 0, when the zygote is formed inside the mother's body, it has to run a clock. If the clock is delayed, then there's a developmental delay. So when an mRNA makes protein, this protein is folded, right? We talked about, when we talk protein, this is alpha helix, and this is beta sheet. So we have talked about proteins before. So protein folding is very important for protein function. So what is protein? Protein is a sequence of amino acids, right? So this is also a sequence of amino acid, but this will not function because this is not folded. But when a protein gets folded, then it acquires alpha helix, beta sheet, or beta sheets, or alpha helical protein. Then folding is important for protein functions. Right? So sometimes what is the challenge? That human proteins may not be folded exactly the same way inside the bacteria. So let's say if I'm expressing a transcription factor, and it has a certain shape inside human body or human cell or eukaryotic cell, that may not happen in bacteria. So that brings a lot of challenge. So a lot of modification has to be done for an adequate functional protein production. Also, such unfolded proteins are degraded inside the bacteria. Cells don't like non-folded protein. They could be toxic to the cells. And it makes sense, right? This is unfolded. These are non-functional. So why it is staying there in the cell? So these proteins are degraded by a system called as proteasomal machinery. So coming back to the same thing, if you want to control transcription, we have to know promoters. So promoters is upstream of start site where RNA polymerase bind. So -10 data region, -20 to -13 upstream. So it is indicated by a minus sign, and -35. So these are the various promoter regions which are important for transcription. So now see a figure here. So you see this, you compare, so let's say this is an SDS page. I don't know whether you have done this experiment in cell biology or not, but this is a picture of a gel. Gel is a matrix in which you can run protein. So this is a molecular weight marker. So the protein gels, they run from heavy to lighter. Now compare the lanes. So this is 1, 2, 3, 4, 5, 6. Compare the lanes 2 and 3, 4 and 5. And this is 50 kilodalton protein, right? Here's the standard molecular weight marker, 50 kilodalton, 50,000 Dalton molecular weight. Now look here, in this lane, there is hardly anything, right? But here you see big blob. What is this? This is protein overexpression. So the difference between 2 and 3 is that IPTG was added here. In the third lane, IPTG was added and the protein expression just scaled up. 4, no IPTG. 5th, IPTG. So these IPTG, they work at the lac operon to scale up the transcription and translation. This is also a very important figure to simplify. If the glucose concentration is low, right, lactose concentration is low, and cyclic AMP is high, then remember, so this is very important question, right? Remember this. This is the mechanism of gene regulation. So if glucose is low, lactose is also low. So why should bacteria spend its currency on transcription? So they should repress it, right? So what happens? Cyclic AMP binds to cyclic AMP activated protein cap. And then lac repressor binds to operator or promoter, no transcription or low transcription. When glucose is high, lactose is low, cyclic AMP is low, again, repressor is there. So here, ideally, there is no repression. Here, there's no transcription or translation. When glucose and lactose is high, then also low. Now you can ask me why. So now see here, when the lactose concentration is high, then it facilitates the binding of RNA polymerase. So this lactose molecule, when it comes, it works on the lac operon, the model I showed you. And then it helps the RNA polymerase to synthesize the gene or mRNA. Okay. So, these are various plasmids that can help in the transcription and translation. Now, some of the promoters, they are very temperature regulated. This is to scale up protein. Analogous to IPTG, you see here how people have engineered a protein. At normal room temperature, mRNA is made, and then the temperature increases to 30 degrees Celsius. So normal room temperature could be 30 degrees. A bacteria could be grown in 37 degrees. But when the temperature comes to 30 degrees, right, there is no expression. And when the temperature is increased to 42 degrees, then there is a active protein production. So temperature regulated. So promoters can be regulated by lactose. They can be regulated by temperature. And IPTG also. Right? So these are some of the examples of protein expression. Now I would come to IL-2. IL-2 is one of the major proteins. It is called as interleukin-2. This is a major protein that helps in immunity, right? So it's possible that some people may be immunocompromised and they need additional IL-2. Now this IL-2 has to be secreted into the human body. It cannot be. So there are two kinds of protein, a protein that can be intracellular or can be secreted. So IL-2 for secretion, it is cloned in a different way so that it can go out of the cells. Now sometimes what happens that when metabolic load, when inside the bacteria, when a plasmid is added for expression of proteins, then this bacteria becomes stress. So increasing number of plasmid leads to limited source of oxygen. Such overproduction of foreign protein also compromises the cellular protein of bacteria. So metabolic load is something that bacterial body reacts to production of extracellular protein and human protein. So that's metabolic load. So with this is a small chapter. What I wanted to communicate, that the lac operin is a model system where bacterial transcription and translation can be used at an industrial level to increase the production of protein, such as IL-2 or any other protein. So with this, we finish this chapter. Do you have any questions? So if there are no questions, then I would like to end this class because I have to meet a student. So I will end the class, and then this recording should be downloaded to my computer. Then I have to convert into YouTube. So it will take me 10 minutes again to come back and start my Zoom. So student who will be meeting me should meet me after 10 minutes. So it's 5:47 now. So let's meet at 6 p.m. And if there are no questions, then we can end this class.
Professor, could you take my attendance?
Okay, yeah. So let me just talk to students who came a little bit later. So so you are. Let me just recall the name: Anderson Hoya, Nafal Bajwa, Bosum Asata. Are you here, Bosum Asata?
Yes, I'm here.
Okay. Then Seguin Brunard, I'm sorry. I saw you coming in. Then Sandra, Dyer Morrison Sandra.
Wait, Professor, what is the red for?
People who are not there. So let me take the attendance, then I come back. Please, recording is also going on, so let me just. Lakisha, Shamika Lawrence, Mirenge, Yel Nakshumo, yes, I saw you. Then Tatiana Ross.
Professor, Kiara's here.
Okay.
Kiara Donald.
Okay. And Anika Thomas, I saw.
I'm here, Professor.
Yeah. Yes, somebody was saying something. Yes, the red indicates absence. Was anybody here and I marked by mistake? Who was that?
Because I see a lot of red from me. I'm always here.
Who may I know your name?
Bernard Sagine.
Okay.
Thank you.
Anybody else? Okay, then I end the class and I come back after 10 minutes. Okay, bye.